Hello, everyone, and thank you for clicking on our poster today. I'm Amanda Ledford.
And I'm Jessie Holstenbeck, and we are with Lee University in Cleveland, Tennessee.
Our bacteriophage that we discovered was Schimmel's 22. It is a member of subcluster EA11. And just to give a little background on bacteriophages, they are becoming more and more recognized as possible medical and ecological uses. So understanding each function of the protein helps with targeted applications such as multi-drug resistant bacteria and bacteria infections in crops.
And as far as our methods, we got our soil sample from Schimmel's Park on our university's campus in Cleveland, Tennessee, and we followed the phage protocol for an enriched isolation. And after purification round and multiple rounds of amplification in order to get a webbed plate, we sent our titer off to the University of Maryland at Baltimore County, where they did a microscopy of our phage. And that was where we were able to discover that we had a Siphoviridae phage with a long tail and a round capsid head. We also sent our DNA titer to the University of Pittsburgh, and they were the ones who sequenced our genome. So within our cluster of EA, we discovered we were in the subcluster of EA11, and there were only two other completed genomes in this subcluster, Hercules XL and IXL. And as you can see here, we have our Famirator map from James Madison University, and we have Hercules XL on the top, IXL in the middle, and Schimmel's 22 on the bottom. And as you can see, we had great protein alignment with our other members, and we had many conserved domains. And based off of Synteny, many of our calls were expected, and we were able to call them with a high degree of confidence, such as proteins like terminase and the tail assembly proteins.
One particular protein that we decided to focus on for this poster is the MASG-like nucleotide pyrophosphohydrolase. It's found in less than 3% of genomes in the EA cluster, and its function is to allow phages to continue to propagate despite cell starvation of amino acids. So when a host cell is starved of amino acids, the starvation alarmone will be made, and it will inject a toxin into the host bacteria cell and cause programmed cell death. So whenever the host cell dies, the phages cannot continue to propagate and live.
So the importance of the MASG in phages is that it increases the survival rate of the phage by preventing the host cell from cell death. Basically, the phage, once the host cell dies, the phage is going to die shortly after that, so it needs it to continue its survival. And just some facts about our genome, we had a length of 39,807 base pairs with a GC content of 63.9%, and we did discover that we did not have any tRNAs. As far as future directions, we are going to continue editing and calling starts with DNA master. We're also going to attempt to answer the question, how does incubation temperature affect the growth of a phage? So is the optimal incubation temperature for Schimmel's 22 different from the temperature that we did use?
And we just want to know the temperature differences because our titer was a little bit lower than we would have liked. So we want to understand if the temperature affected that low titer concentration. We would also like to know the host range of our bacteriophage compared to other bacteria besides the M. filarium that we used. So would another bacteria, say E. coli, match with Schimmel's 22, and would it allow for greater growth of our phages?
And we just wanted to say thank you to Lee University and the Phages program and HHMI for letting us conduct this project.
Thank you.